Going into a store to look for baker's yeast can be a dilemmatic experience. Let me explain. Before I really got serious about baking, those rows of baker's yeast look pretty much the same to me. After all, it's just one species, right? You just pick up a bag and go make your bread, or in my specific case, fermented honeycomb cake. But then came the failed batches of cake, and eventually it dawned on me that my yeast was terrible for this application. It wasn't doing well surviving in a batter with lots of sugar. You might relate if you tried making a sweet bread recipe and found that the yeast just wasn't rising. Stressful times. But digging deeper, there are so many types of baker's yeast. What is the difference between fresh yeast, active dry, or instant yeast? And which one is best for me? What about all those different brands? Is there a good reason to choose one over the other? Aren't they all the same? And with all the research going into it, how do we know if our yeast isn't genetically modified? Are these genetically modified organisms? Well, that's what this video is for. We're going to be delving into the science of baker's yeast, as well as what those additives on the ingredients list really do. We'll also be looking at why you shouldn't proof instant yeast, why it's important to get the right baker's yeast, and more. By the end of this video, you should know everything that you need to know about baker's yeast. So let's jump right in. Okay, we've established that there are different types of baker's yeast. Now let's delve into the two broad categories that the baker's yeast is typically sold in: fresh and dry yeast. Many savvy bakers will tell you that fresh yeast makes bread that tastes way better than dry yeast, and they could be right. But it might not just be the yeast. Rather, it could be the lactic acid bacteria inside the fresh yeast. Just like in sourdough, these lactic acid bacteria could be responsible for producing more flavorful aroma compounds in the bread, and some brands of fresh yeast may have more lactic acid bacteria in them than others. So the people who say that fresh yeast makes better bread than dry yeast alone are probably picking up on that. But that doesn't necessarily mean that fresh yeast is the best option for you, because although it's got more living yeast cells, fresh yeast typically has a much lower storage life when compared to dried yeast. Fresh yeast should be refrigerated but not frozen, and it's best used within several weeks of purchase. As a home baker, to be honest, I rarely use fresh yeast. It makes more sense for me to use a bit of sourdough starter or fruit yeast water for added flavor. Now, as for dry yeast, it's often sold as either active dry or instant yeast, with the difference being that active dry yeast requires time to rehydrate, normally in the form of proofing or blooming, while instant yeast does not. Okay, this was something that always confused me before I dug into it, but it turns out it's tied to the fact that instant yeast has a finer granulation than the coarser active dry yeast. This allows instant yeast to have a higher dispersion and faster rehydration. This is part of why instant yeast doesn't need to be activated or proofed before you use it. In fact, doing that might be a bad idea. Some studies have talked about how since instant baker's yeast is so fine, these cell constituents can leak out during rehydration, and that it could end up affecting the leavening properties of the yeast. So it may actually be safer for you to just throw in the instant dry yeast into the flour rather than activate it. Across the board, most yeast producers recommend doing this for instant yeast as well. Admittedly, in some of my old videos, you can see me adding the yeast into the water. Well, mea culpa. In terms of viability, active dry yeast has a shelf life of up to 2 years under an inert atmosphere, so an airtight bag or container, and it can be stored at room temperature. Instant dry yeast has a similar shelf life of about 2 years in a sealed package, but after being opened, it might be more susceptible to oxygen due to the larger specific surface area, causing it to have less viability. All in all, if you want your dry yeast to last, regardless of the type, keep them away from the air and high temperatures. I personally mainly use instant dry yeast, and I keep it in the freezer in an airtight container. It's served me well, and I can generally get through a bag without worrying about my baker's yeast failing to leaven anything. As a bit of trivia, instant dry yeast is a relatively more recent innovation as far as dry yeast goes. According to one paper on dry yeast patents, the first patent for an instant dry yeast that didn't need rehydration was assigned in the US in 1957. Now, in this very patent, we can actually see an ingredient that is still in common use today. You may very well find it on the ingredients list of your package of dry baker's yeast. Sorbitan monostereate is an emulsifier that is applied to the baker's yeast before it's dried. It's typically added at up to 1% of the dry yeast's weight. One of the things it's said to do is help protect the yeast while it's being dried, and it also helps during the rehydration of the yeast cells when you mix the dry yeast into the bread dough, as yeast is naturally hydrophobic. Sorbitan monostereate remains one of the most popular options for this application today. Another common ingredient you'll see added to the yeast is ascorbic acid, commonly known as vitamin C. It's added as a dough improver. It strengthens the dough and also improves gas retention while giving the bread a softer and finer crumb structure. Now, delving a bit deeper into the science, you should know that ascorbic acid by itself isn't really what strengthens the dough, but inside the dough, due to a process involving oxygen, ascorbic acid readily becomes dehydroascorbic acid, or DHA, which is thought to be the real dough strengthening agent. That being said, because the conversion from ascorbic acid to DHA relies on oxygen, if the oxygen in the dough runs out, likely due to the yeast using it up, then there is a chance that the ascorbic acid will actually weaken the gluten structure because it's not being turned into DHA. So long fermentation with ascorbic acid inside the dough is generally a bad idea, and you need to make sure to choose the right yeast for the dough. This may be part of the reason why we tend to see ascorbic acid added to the yeast for sweet doughs. Since many sweet bread doughs typically have shorter fermentation times, they're less likely to deplete the oxygen needed for ascorbic acid to convert to the strengthening DHA. The addition of ascorbic acid in this case could also be due to the fact that sweet bread doughs often call for larger amounts of yeast to be added. You might wonder, what does that have to do with adding a dough strengthener? Well, large amounts of yeast, especially dry yeast, also means a larger amount of dead yeast cells. When yeast cells die, their cell membranes become porous, allowing their contents to leak into the dough. What we're interested in is the released glutathione, which interferes with the gluten network, ultimately weakening the bread dough. This is why more dry yeast, or just more yeast in general, generally results in a weaker dough. Ascorbic acid is added into the yeast to help counter this and strengthen the dough. But there are always two sides to a story. While glutathione isn't normally a good thing in sweet bread doughs that already have a lot of fat for extensibility, it can be a good thing for things like pizza doughs because glutathione allows the dough to stretch out more easily. That's why dead yeast is sold as an additive. It contains a lot of glutathione. You could get the same effects from some nutritional yeast. Now, let's talk about the yeast itself. Whatever baker's yeast might have started out as, after 10,000 years of human domestication, the species has undergone a lot of adaptations. If you'd like to know how the baker's yeast is produced, fresh yeast is made by removing extra water from the purified yeast that's been propagated in large vats, and compressed fresh yeast can have moisture contents of 60 to 75%. That is a lot of water compared to dry yeast, which goes through an additional drying step, where the yeast is extruded into thin ribbons before being dried using special equipment that blows heated air through it at moderate temperatures. Not high enough to kill the yeast cells, obviously, but enough to dry them down to somewhere around 5% moisture content. Then the dried yeast is vacuum-packed in heat-sealed nitrogen or carbon dioxide flushed foil packs, forming hard, airtight bricks. For the yeast to even survive these processes that allow us to get them in this dry form, they are already selected yeast strains that can survive very harsh conditions. For context, a fried potato chip has a moisture content of around 2%, and a raisin typically around 15%. So the 5% moisture content of dry yeast is really spectacular for a living organism. So these yeast strains are definitely the cream of the crop in terms of surviving. And in fact, a lot of the variety in yeast sold is basically about just that, survivability. Consider these two bags of yeast. One is designed for sweet doughs like brioche or Asian bread, and the other is recommended for lean doughs like baguettes or artisan breads. If we use the yeast for baguettes in a sweet dough recipe, we would see firsthand how difficult it is for the yeast to survive in such a sugary environment. It would ultimately result in very long fermentation times and greatly stunted leavening ability. This lethal stress that the yeast goes through is called osmotic stress. The simple explanation for what's going on here is that there's just so much sugar in the surroundings that it results in the water inside of the cell being sucked out because of osmosis. Water moves from areas of low solute to areas of higher solute. And since there's so much sugar, a solute, outside of the cell, the water goes out. This is a rapid process. The yeast cell can literally shrink in a matter of seconds when exposed to severe osmotic stress. Salt can also exert this type of osmotic stress on the yeast, although surviving high salt levels is quite different from surviving high sugar levels. But we'll focus on sugar and sweet bread doughs here. As an added factor in sweet doughs, when the yeast breaks down the sugar to eat it, it actually increases osmotic stress because the sucrose turns into glucose and fructose. Many brands sell yeast that are specifically geared to survive and thrive in these doughs, called osmotolerant yeast. And they're normally differentiated from other types by the color of their packaging. Here, there are 3 options, one for lean doughs, one for moderately sweet doughs, and finally one for very sweet doughs. The yeast for sweet doughs are naturally the osmotolerant yeast strains. These yeast cells are prepared to face an environment with plenty of food since they can break the sugar down with the enzyme invertase. However, they must also survive the osmotic stress. To counter this, these yeast strains accumulate lots of osmolytes that help regulate osmotic pressure, especially glycerol and trehalose. Going into more technical detail, glycerol helps by balancing intracellular osmolarity, making the interior of the cell resemble the environment a little more. It's called a compatible solute because it doesn't interfere with respect to intercellular processes while also minimizing the effects of osmotic stress. Going into the cell mechanisms, inside the cell, glycerol is produced by a 2-step pathway that involves 4 genes. The first step relies on GPD1 and GPD2, while the second step requires GPP1 and GPP2. This is basically the only route for glycerol production in yeast. Studies have found that the deletion of either the first pair of genes or the second result in complete abolishment of glycerol production. This shows the importance of these genes in producing glycerol for the yeast, and increased expression of GPD1 has been shown to result in more glycerol production in the yeast cells, which in turn has beneficial effects on yeast adaptation in doughs with high sugar content. In one study, researchers compared 2 yeasts used for sweet doughs to 2 yeasts used for non-sweet doughs. They found that these sweet dough yeasts had efficient expression of GPD1 and produced more glycerol than the non-sweet dough yeast did. Naturally, the yeast for sweet doughs also survived better under high sugar. Now, one major influence on GPD1 is the so-called HOG pathway. Not that HOG. HOG stands for high osmolarity glycerol pathway. This pathway is activated when the yeast cells sense osmotic stress, and in response, it releases the cellular equivalent of signal flares that tell the yeast to stock up on glycerol, among other stress response actions. More specifically, the HOG pathway increases glycerol accumulation by inducing increased expression of the biosynthesis genes that make glycerol, preventing the release of glycerol from the cell by closing the glycerol channel FPS1, and increasing the expression of STL1, which could contribute by importing more glycerol from the environment. So let's see the HOG pathway in more practical terms for us bakers. I have one study here that looked at yeast in bread dough fermentation and found that the deletion of the HOG1 gene and other related genes in the pathway resulted in significant reduction of fermentative capacity. So the takeaway is that these genes are something that are greatly needed by our baker's yeast to produce good bread. And naturally, this pathway is likely to be very important for osmotolerant yeast strains. Quoting from the paper, Mutations that inactivate the Hog pathway make yeast highly sensitive to hyperosmotic stress, which is definitely a bad thing for yeast living in sweet dough. However, there is a trade-off, since too much Hog pathway activity is lethal to the cell, and for that matter, so is too much glycerol. Problems with the FPS1 channel, which controls glycerol release, can lead to excessive glycerol buildup within the cell, resulting in poor growth under anaerobic conditions and cell fusion defects. So for the yeast cell, maintaining glycerol levels is a balancing act, and its survival in many conditions relies heavily on its ability to rapidly regulate these levels. Now, let's talk about trehalose, sometimes called the sugar of life. This natural sugar is made of two glucose molecules, and it's been linked to more than just osmotic stress tolerance in yeast. Trehalose accumulation has been linked to better growth under ethanol or alcohol, as well as heat stress, and it also helps the yeast survive better under freezing stress. Some of the genes associated with trehalose in yeast are the genes that produce trehalose, and the genes involved in the making of trehalases, which are enzymes that break down the trehalose. In general, if a yeast strain has more expression of the genes that make trehalose and less so of the genes that break it down, then it probably has more trehalose, although, of course, the real genetics of the situation are much more complex. Outside of yeast, I've actually baked with trehalose as an ingredient and tasted it on its own. It's got a very refreshing taste on the tongue, and it's supposed to be much better for your teeth than sucrose. Due to its specific properties, it can allow for the making of bread that's perfectly soft and sweet without a crust. While we won't go into it in this video, we made this video all about it, so do check it out if you're interested. But anyways, beyond just surviving osmotic stress in high sugar doughs, both trehalose and glycerol have also been shown to help these yeasts survive in frozen doughs, increasing their cryotolerance. So if you're thinking of freezing your bread dough, it may be a good idea to pick up some osmotolerant yeast. But then again, it's important to remember that picking the right yeast goes beyond osmotolerance. So let's look at the yeast made for lean bread doughs. Here, the yeast are designed to live in an environment where there's no severe osmotic stress, but there is still a challenge. You see, baker's yeast has certain preferences in terms of food, more specifically the type of sugars it likes to eat. Glucose and fructose are some of its top favorites, and then it breaks down table sugar and fructan easily with an invertase enzyme, so those are gone pretty quickly too. And it's only after it has practically depleted all of the above sugars that the yeast will finally go for maltose. This poses a problem for lean doughs because maltose is the most abundant sugar in these doughs, and so the baker's yeast inside the dough quickly runs out of its preferred foods. The system regulating all of this is called glucose repression. For us bakers in lean bread doughs, we want the yeast to quickly lift glucose repression and start eating maltose. Yeast strains that can't do this quickly enough enter a long lag phase where the fermentation rate drops rapidly along with the carbon dioxide production. Practically speaking, a lagging yeast in a lean dough would ferment well for about the first hour or so until most of its preferred sugars were almost completely degraded. However, as the yeast struggles to adapt to maltose utilization, gas production stalls or slows down significantly. This can negatively impact dough rise and might lead to suboptimal bread. This difficulty in adjusting to maltose consumption is more pronounced in wild baker's yeast. Wild strains of baker's yeast are usually unable to or only very weakly utilize maltose. This may be due to the fact that maltose is a relatively rare sugar source in the environment of these wild yeasts. To even be able to use maltose as an energy source, yeast cells need to have certain genes, more specifically, one of 5 unlinked mal loci, loci being the location of the genes on the chromosome. Each of these spots on the chromosome or each locus contains 3 genes that are used to process maltose. One gene encodes a dorman for the maltose, letting it into the cell, a maltose transporter enzyme. Another gene encodes an enzyme that breaks maltose down, a maltose hydrolyzing enzyme. And the last gene regulates the previous 2 genes. The first two genes correlate directly to the process of the yeast eating the maltose, because unlike with table sugar, maltose consumption requires that the yeast bring the maltose inside the cell using a maltose permease and break it down there with a maltase enzyme. We can generally infer that the yeasts specifically sold for lean doughs have these genes, possibly even in multiple copies, as observed in many strains. It has been speculated that a higher number of these genes in a yeast cell correlates with its ability to consume maltose, making it particularly beneficial for lean doughs. So this is another good reason to pick up the right bag of baker's yeast for your dough. Although an osmotolerant yeast strain may be highly tolerant of sugar, it might produce denser bread with less volume in a lean bread dough due to its trouble adapting to the maltose. But what if a yeast strain could excel in both areas, if it were both osmotolerant and capable of utilizing maltose to the highest level? Would the creation of such a yeast strain further simplify our choices? You wouldn't need to search for a different yeast for your sweet cinnamon rolls or your French baguette, just one type of yeast to leaven them all. Interestingly enough, a strain capable of this already exists, and it's been around for quite a while. In a 2001 paper, researchers aimed to develop a strain that leavens both sweet and unsweetened dough. They succeeded in making this strain without resorting to genetic modification, a topic we'll explore further later. But it's been more than 20 years since this paper, and we still see osmotolerant and non-osmotolerant yeast strains for sale, along with a whole variety of yeast marketed for specific purposes. Perhaps it's already available under a multi-purpose label, and there could be other reasons why different baker's yeast for sweet and lean doughs persist, such as limited osmotolerance, impractical production methods, or other factors. So now that we've discussed yeast for different kinds of bread dough, I want to briefly talk about the more subjective aspect of flavor. Although many people don't consider it, yeast can play an important role in shaping a bread's flavor profile. Different yeast strains can produce different flavors, as shown in a study examining the flavor profile of breads produced from various yeast strains sourced from the baking and beverage industry. These beverage yeast strains included those used to make wine, beer, and ale. Researchers found a pretty wide variety of responses on how the bread tasted. Some strains had a cheesier taste, some were buttery, and some were bitter. Interestingly, but also unsurprisingly, the baker's yeast strain associated with baking produced the least bitter bread. The genes responsible for aroma formation by yeast have been found to belong to the Ehrlich pathway, which essentially turns amino acids into a variety of alcohols. So differences here can definitely affect the flavor of the bread. Considering this research, it wouldn't be surprising if there were subtle differences in flavor from different yeast brands, and why some people might prefer the flavor of one over the other. You might wonder, but aren't all these brands using the same strains of baker's yeast? Well, not necessarily. It turns out companies can actually patent yeast strains. Under the so-called Budapest Treaty, a company can deposit the strain to an international depository authority, which will store the yeast, and then the company can patent it. This patent will be recognized internationally in more than 80 countries around the world. As of 2024, there are 50 such authorities that accept microorganisms, 7 in the UK, 3 in the US, China, India, Italy, and Poland, as well as many more scattered around the world. So a company can very well protect a strain from being produced and resold. And now we can talk about how strains are produced. Just like we asked in the opening, is there genetic modification at play? Well, for context, you should know that genetic modification of yeast is already an established field with countless papers documenting it. I'm not saying that it's good or bad, but genetically modified yeast is of interest to many non-food related fields as well, including the biofuel industry and the medical industry. Now, in baking, many yeast brands have stated that their yeast strains are non-GMO, so if you prefer not to consume genetically modified organisms, you can mostly rest assured with your yeast. However, non-GMO doesn't necessarily mean a natural culturing method like in sourdough or things like old dough. There are a whole range of techniques that fall under non-GMO that still involve quite a bit of science, such as the use of UV radiation to induce the creation of mutant yeast cells. These mutations are typically random, and most of them will kill off a large amount of the baker's yeast, but some mutations could potentially lead to certain desired traits. After several rounds of selection and mutation, scientists can eventually come up with a strain that has the desired phenotypic traits. This technique is called mutagenesis, and using UV radiation as a mutagen, it's not typically considered a GMO technique for microorganisms. There are other mutagens, and many aren't considered GMO techniques as well on microorganisms in general. This makes sense because mutagens create random genetic changes by stressing the organism, unlike gene editing, which can target specific genes. Mutagenesis is generally employed along with techniques to breed the yeast, including direct mating, mass mating, and protoplast fusion. Direct mating is the most intuitive to understand, just like how we breed animals by putting two together. Direct mating literally means just putting yeast from two carefully chosen parent strains together and letting them mate to produce offspring. There are some variations because we're talking about yeast, but that is the general principle. Mass mating is similar, but this time more yeast and probably more parental strains are used, allowing for many crosses to happen. It basically quickly results in many kinds of offspring for scientists to look at. Now, protoplast fusion is quite different from the two previous methods. Quite literally, it is the fusion of two cells together. This is done by removing the cell wall, inducing hybridization, and then rebuilding the cell wall. The idea is to get two cells with the desired traits, combine them, and get a cell with the combined desired traits. It doesn't necessarily have to be done within a species. Often, a baker's yeast cell will be combined with an entirely different species to create a cell with certain traits, such as the ability to eat lactose, temperature tolerance, and more. Protoplast fusion is also useful for yeast cells that have trouble mating, that can't sporulate, have inviable spores, or other issues. Another popular technique for creating new yeast strains is adaptive or directed evolution. It's funny to note that while we have so many techniques for creating genetic diversity in the yeast, baker's yeast itself can have a very fast evolutionary rate when put in the right environment, with adaptive changes happening every 40 generations. The idea of adaptive evolution is so that the yeast eventually adapts and evolves to survive their surroundings. So essentially put them into certain conditions and let them survive over many generations. This is normally used to fine-tune a trait that a yeast strain already has, though. Now, all of the techniques above have been used for many applications, including the production of yeast that will better survive in frozen doughs, yeast that will better ferment chocolate, and yeast that will make better beer as well as wine, and more. It is no wonder that this small microorganism is ubiquitous, and it's really quite amazing that we can have easy access to such an incredible microorganism. And that's it for this video. Thanks for watching, and bye!